Yeah, I guess we can get started, but as I mentioned, this will be more conversational. So any jump in at any time with any questions, comments, insights, as we'll kind of go through. We have domain expertise in the inoculants, in the analytics, and all of that, but you guys have that domain expertise on the turf front. And some of our conversations, Jake, have provided a lot of insights. So that will help kind of guide this conversation. Thank you for making the time to meet with us and go over some of these results. So most of you, a few of you have met me, but my name is Michael. I'm a scientist focusing on microbial ecology, so just how bacteria, fungi, everything interact in the ecosystem. And then Donya and David are on the call as well, and they've done a great work organizing, coordinating you guys, which has been very helpful. So in terms of kind of this domain expertise that we bring to the table, it's this bioprocess optimization. You know, the typical approach is turf core samples, get physiochemical data, those results get analyzed, and then you have data-driven decision-making in your turf management. And we help growers with this broad spectrum biological or microbiome side of this equation. So things don't exist in a vacuum. The biologicals are doing things in the environment, which we'll talk about in a minute about ecosystem services. So we help these growers with that microbiome and disease detection for their crops. And we do this by using the latest genomic technologies and these statistical or data-driven insights that help early pathogen detection, remediation, crop loss prevention. And a bit of a background, what we're talking about when we say plant microbiome, that is just the bacteria, fungi, eukaryotes living in, on, or around plants. So the same way we talk about, you know, the gut microbiome in humans. You know, these are just the bacteria mostly or fungi that live either in the rhizosphere, which is just below the surface, the above ground phyllosphere, or the endosphere, which is inside of the plant itself. And that is the site of action of our inoculants. And turf, these live organisms are inside the plant as endophytes. And there's millions and millions of bacterial species, and grasslands in general tend to be quite diverse. There's tens of thousands of species in any given site. Managed systems like golf courses tend to have lower diversity. They tend to have, you know, less litter inputs. You only have a small number of plant species, so they're shedding a lot less material into the ground, and there's no tillage or things like that. So these managed systems tend to have lower diversity, which can make the plants more susceptible. So a good way of combating that is fostering diversity. And our approach is to use DNA-based microbial profiling. And there's two approaches. You can take an amplicon sequencing, so an approach that you basically make a barcode. You look at all the species by looking at one gene that every species has. And then you can look at what they're doing and where you sequence every gene in the sample. And most of our analytics right now in this talk is going to be about who is there. So we can screen for all living organisms in a sample of soil. So we look at all bacteria, all fungi, that type of thing. And that gives us insights into nutrient cycling and pathogens. We have very early detection of pathogens. And it also gives us insights about what's missing in a green, in a turf sample. You know, is there low population of bacteria involved in nitrogen cycles? So do you need to supplement with nitrogen? So it is a data-driven approach to management. So the project that we'll talk about mostly is this Polo cub case study, where there were 3 greens with different types of amendments, and the goals were, do microbial communities differ after the application of our product enter? Are these differences, are there differences between different greens? So do the differences correlate with the health or performance of these greens? And are there any notable pathogens, things from fungi, bacteria, and water molds, in these samples, and how do these pathogens respond to inoculation of enter? And early on in this project, we saw on the 18th green, 4 days after the first application of N-turf and other amendments under Celebration Bermuda, there was a pre-application of visible drought-stressed patches and thinning. And after one application, there was a notable rapid improvement in things like turf color and density. And this is indicating an enhanced root zone activity, water and nutrient use efficiency, and things like that. And what does N-turf do to turf? Has these 2 endophytic bacterial strains, 2 bacteria strains that live inside of the plant. And each of them are doing some key metabolic functions that have been proven to be effective in Gulf turf systems. There is improved disease management. So there's an inhibition of mycelial growth, some key fungal pathogens. This is done by several methods. One, for example, is depleting nutrients essential for pathogen development. So it's out-competing. There is improved nutrient uptake and stress recovery. So these organisms, these endophytes, actually help nutrient bioavailability. They make nutrients more bioavailable to the plant. So it facilitates uptake of nutrients. And one of these, Stutzeromonas, has biological nitrogen fixation. So it's converting this atmospheric nitrogen into plant available ammonia. So in nitrogen limited conditions, it will supplement nitrogen available to the plant. So I'll go through a bit of the results from this broad spectrum sequencing, which covered all bacteria, all fungi, and water molds. On this image, you can see that the blue is from, so the batch 1 in red, that is pre-application of amendments. Batch 2 is 4 days after, and batch 3 is several months later. So it's late in the process. So we have an increase in biodiversity. The higher in this plot, the higher on the y-axis, that's the higher biodiversity. There's more species and they're better distributed. So they're increasing over time. This biodiversity increases over time after inoculation. And what does that do? That improves ecosystem services. Bacteria are doing a lot of things in soil. They're improving nutrient bioavailability, as we mentioned. They're releasing, they're degrading litter and turnover in the soil as roots die off. Bacteria and fungi are making that carbon, that nitrogen, those nutrients available to the plants again. The more diverse your community is, the better that ecosystem functions. This is called ecosystem services. So instead of management having to supplement all of these nutrients or managing the ecosystem, a lot of the ecosystem services are being done by a diverse system. Biodiversity also increases resilience, stability, and resistance. This means resistance to change, but it also means the ability to respond to change. You have disturbance, you have a drought, for example. The more diverse a system, the better it is to return to a stable state after the disturbance changes or is over. Biodiversity tends to be higher in the plant than soil, which is consistent with turf management. This isn't always the case in general grasslands, but you have a low litter input, so the soil itself community has a bit less diversity than you would in a grassland. But this is consistent with your management system of the system. One good thing is the biodiversity we're seeing in this site I don't know if it's a general rule, but in this study, the biodiversity we're seeing is typically higher than other managed systems. So you do have a relatively diverse system compared to other managed systems that we work with. When we bring this over to fungi, similarly lower than natural systems, fungal biodiversity, and this lower fungal biodiversity is almost exclusively due to grass being essentially a monoculture. You don't have a lot of leaves being turned into the soil. You don't have a lot of things, but your system is diverse as it needs to be. So fungi don't respond as quickly to something like an inoculation. You're encouraging biodiversity in the bacteria. Fungi tend to be slower to respond, whereas bacteria, you see a change really quickly. Shorter life cycle, they grow quicker. So this comparison is just comparing the communities at a site. And we're only going to talk about the bacterial one because that is the one that is experiencing the most rapid change in response to application. All you need to know here, the colored dots represent batch 1, 2, and 3. Again, red is 1, green is 2, blue is 3. And the closer 2 points are together, the more similar those communities are. And what this analysis is saying is that roots and plant communities are very different. So up in the top right quadrant, you're seeing a lot of the plants from batch 1 and batch 2, and you see rhizosphere on the left quadrant here. So roots and plant communities are very different, which is what is expected. The endophyte communities are changing over time, so what is being brought into the plant is changing over time in the same way. So roots and inside the plant system is more similar at a batch, and they're changing over time together. But what is really interesting is by batch 3, your endophyte and rhizosphere, so inside the plant and around the plants, communities are starting to be more similar. All of the blue samples, the rhizosphere and the inside the plant, are becoming more similar to each other. So you're seeing a bit of a change over time. We have a couple of notable things before we get into specific species, but Xanthomonas, which causes diseases like bacterial leaf streak, wilt, water-soaked streaks in grasses. They were dominant. It was dominant in pre-application samples, but kind of tapered off after application. And that could be because of the application, or it could be environmental, because these, weather is changing. The nitrogen cycle is strongly represented in these samples, which makes a lot of sense. The ANPR group, that's just rhizobiales, like rhizobia, different bacteria that are involved in nitrogen cycling. So I'm going to give 3 good examples of tracking individual groups of organisms in the samples. NTERF is composed of Pseudomonas and related organisms. And what we're seeing over time is there's an increase in Pseudomonas diversity and abundance as we progress through batches. More diversity by batch 3. Batch 1 and 2 have less diversity than batch 3, although the abundance is slightly going down by batch 3. Pseudomonas is higher in plant roots on average, which makes sense. They are endophytes. And we do have qPCR detection of all NTERF species in most samples after inoculation. So batch 2 and 3, we have detection of NTERF in rhizosphere and plant samples. Detection is low. They are members of a fairly complex community, but they are present, the species that are involved. We also found an interesting note here that we have a really high abundance of cyanobacteria in some samples. So after a post-drought sample, so after drought with increased rainfall, there was a spike in cyanobacteria, especially in shaded areas that stayed moist longer. And we can, using these tools, determine risk factors for cyanobacteria algal spikes in samples. Things like pre-existing species or environmental factors such as tree cover and nutrient cycles that can prime your system for a bloom in cyanobacteria. So if you have a very low probability of algal blooms or blue-green algae cyanobacteria on your grass surface because of your system isn't primed for it, you don't have to manage it as closely as another site which we can tell is primed for that type of spike.
Hey, Michael, real quick.
Yeah.
Since we discussed that in the previous meeting for the cyanobacteria, that was one thing that you raised to our attention was that we believe that the majority of the issue is coming from water. And since we know that the cyanobacteria causes this algae on living surfaces, we actually increased our hydrogen peroxide applications to our greens. And I've seen a huge reduction, especially going into these lower light months, a huge reduction in surface algae on our greens, where we didn't have to apply it other algae fungicides out there. We were actually able to control it by doing more hydrogen peroxide applications, knowing that information going into these lower light months.
That's great. That's yeah, exactly kind of
Sorry, Michael, could you just repeat that? I couldn't catch all of it because Jake was kind of cutting in and out. Yeah.
Yeah, in response to kind of knowing what to look for, these cyanobacteria, Jake had an intervention of, I think he said hydrogen peroxide, but an intervention specifically against cyanobacteria. And this targeted approach has led to a significant reduction in that appearance of the algae and cyanobacteria on the surface. And I believe it affected other interventions that were not necessarily required because it turned out to be cyanobacteria driving that change in the surface. I believe that's kind of summarizes what you said, Jake.
Yeah, sorry, my internet connection might be a little spotty in the metal shop. Please let me know if I need to repeat anything.
I think I caught most of it. Hopefully I reflected what you were saying. But yeah, it sounds like it was, yeah, the directed intervention based on these results seem to be effective.
Yep.
Good. Which is one of the strengths of this approach is that we're not targeting individual, we sequence everything. So if it is a bacteria, a fungus, or a water mold, and it is not vanish in the air, we will observe it. We will detect it with this approach. It allows us to do a community first or community driven approach to these data analytics. And you understand how many species are interacting and what systems are primed or what samples or greens are primed for a response that you can manage directly. So it makes interventions much more efficient. One example, another one that I always like to bring up is oomycetes. So these are water molds. They're a group of organisms that contain some really aggressive plant pathogens. Things like Pythium, so damping off and root rot, Phytophthora, so late blight, collar rot, like Phytophthora is the Irish potato famine, that type of thing, like these really devastating pathogens, Plasmoporas, downy mildew. So we have a non-invasive, this approach is sensitive, non-invasive monitoring where you don't require a predefined set of pathogens you're looking for. You're not sending this in for a Pythium test. You're sending this in for an everything test. So this allows early detection before outbreaks. You don't have to wait for symptoms. You can determine if your samples are primed. Your greens are primed for this response, for example. So we have a strong detection strategy, and here we do detect these water molds, these oomycetes, but they're at really low relative abundance. So they're not dominant in the system at this time. But you can confirm if a system is primed for a Pythium outbreak once conditions change. So if you know that this type of pathogen is not present, then no matter what the conditions turn out to be, you're not likely to have a big bloom of these organisms. But if your system has your Phytophthora, your Pythium ready to go, and then conditions are beneficial to that organism, it will take off. We have this early detection before outbreaks in a non-invasive type of monitoring. So that's kind of the key I want to bring out, is this non-invasive monitoring of the entire community. So it allows this early detection of pathogens in more or less real time. Once we receive samples, there is a quick turnaround between receipt and results. Targeted tests on the order of 2 to 4 days. These universal tests on the order of 1 to 2 weeks. And now that we have a lot of shipping, and Jake can attest to this, shipping kind of changed over the last 6 to 8 months. But we've resolved some of those to make shipping across border really easy. That is why our third sample is not a month after application, but several months after the first application. It also allows input timing. So your management decisions being informed by biology, not necessarily a strict regimen or calendar or response to environmental conditions. You get you have the environmental conditions plus the biology plus the nutrient data all working together, targeting your interventions when and where they're needed. And this does extend fungicide and fertilizer efficiency. You are not applying these things when they're not as needed. So you have less input costs and more informed and regulated input costs. Taking advantage of giving you all the information you need to make the best decisions on your end. I think we can just have a discussion now. So thank you very much for listening. And yeah, we can have a discussion about anything we've covered or any other questions you have.